Okay, so welcome everybody. I think the first thing we should really decide is when we do the blog courses. Unfortunately, you can basically see the form responses here, and it's basically like last time. It's like every date has like 3 to 4 responses, so I have no clue how to do this best. But I think the one thing I saw when I went through the responses is that basically either the people have time in the later dates or the people have time in the earlier dates. So I think there's probably no way around to do again like two courses of 1 week, which is also okay because we are a smaller group of like 4 to 5 people per course, which is I would say much more productive, or basically I can help you much more. It's often difficult, like if 10 people often have like 3 or 4 people have problem at one time, like with 4 or 5 people, like it's 1 or maximum 2 people who have problems, and so it's much easier to supervise and do things. So I'm really leaning towards doing 2 courses again, and one in August and one in July. And the big question is, okay, when to do the dates? The big question is for, I'm not sure if Jonathan or Luca are here, if they could really if they can do on one other date, like here or here, then we could basically just select one of the either the 7 to 11 or 14 to 18th, and then we have to figure something out here in the beginning. The problem is, yeah, we have basically four people for the 3rd to 7 and only 3 for this. So I yeah, basically from the democratic vote here, I have to, or I should take the first one, and I have no clue. I would just select one of the both here. No clue. If either Jonathan or Luca are here and there's any way to do one of the other weeks, it would be great. Like I said before, even if you can't do all the days, if you miss one day, but you can do the day afterwards and so on, we can find some flexible solution. It's more like, okay, you should have most of the week, you should participate in the course. I know it's always difficult to find continuous time for that, but okay. Somewhere I have to make a decision here. I don't know. Any strong opinions, or otherwise, okay, I will take this here, and okay, and I would say the main problem is for Amanda and for Jonathan. Well, let's see who's here today. So Jonathan is definitely here. Luca is not here, so yeah, then I will okay, sorry, but then we'll take the later date. It's just, okay, you're here right now, and this makes more sense if you participate, or at least I saw you participated and contributed already, and definitely this is for me it's definitely an important sign. So okay, so the main problem will be here, the Luca and Anderson. Okay. Can you still hear me, or did I lose connection? I'm not sure. Somehow, okay, perfect. Somehow the camera and so on is hanging here. Okay. So just let me know if you don't see anything and so on. So the dates will be 3rd to 7th of July and 14th to 18th of August. Feel free to switch if you can do it, but I can't offer more courses than 2 weeks. This is already a lot of time. Okay. So what we will do today, is mainly 2 things. The first one, I will talk a bit about reproducible research and why this is important and how this is related to our project. And the second part is talking about SBML, which is the systems biology markup language. And this is basically an XML-based format to encode computational models. And We will use SBML mainly for developing the models and running the model simulations. So I think it's really important to get a basic understanding of what SBML is, how it allows to describe such computational models, and how we use it here in this project. And so I think both of these topics are really important as a background, and also to understand why we do or why we develop the models and the data in a certain way. Quick reminder, what we're doing here, we're basically developing a physiological-based pharmacokinetic model of lisinopril. And there are, I would say, like two big pieces which we have to solve. The one is like actually developing the model. So I will go a bit into this today with SBML. The other part is really getting the data and the data together with the model, which is what we will talk more about next week. And then you will also start basically on working on your own publication and getting to know like the first data sets which we will use for the model development. Okay. So short motivation, most of you probably don't know that, but a lot of the biomedical research has a big reproducibility crisis. And what this means is especially like that science goes very wrong in many ways, and one of the biggest problems is really that many of the results which are published, they are just not reproducible. And this means basically if other people are trying to redo the work and try to build on that, they just figure out, okay, this is basically these are not true results. And then this is really a big problem because especially also if we want to build computational models based on data which is reported in the literature. Okay, if we can't trust this data and if we can't trust the reported findings, it makes it really difficult to build computational models, because it's always you have to ask yourself, okay, is this true or not? And is this a fact or not? And this is something which is not much communicated. So today, it's more like if something is published in a scientific article, it's somehow like a truth. But in reality, this is unfortunately not the case. And so this is basically here, like in this nice picture. Okay, if basically a second scientist tries to do exactly the thing one scientist did, and often like just from the information which was provided in the publication, it's very difficult to get the same results. And we see this a lot, like in scientific media. So if you just basically read scientific journals and scientific blogs and so on, there's a lot of discussion about that. And there's like I say, lots of biases, lots of reproducibility crisis, problems like with open data, problems with metrics, and overall we have often very low confidence in preclinical research and in biomedical research overall. And this is I think the public is not really aware of that. It's more like if you actually try to work with data and try to do a research project which is building on data, it's the first time you're getting into contact with this problematic. We see this also in the public media. So like in the last years, this was taken up like by I don't know, New York or other journals and so on, Economist, New York Times, and but also many German magazines and so on. And it's basically like this questioning of science itself. And then this is also like an important thing, like also like not only for biomedical research, but I think for climate change, climate crisis, and like basically a lot of things like in this context of fake news and so on. If we can't trust like, okay, the science and the scientific results, then the question is, okay, what is true? And if we can just cherry pick what we want, okay, what is on what should we build our beliefs? And this is, I think, a much bigger question than building computational models, but it's the same problems apply to a computational model, because you basically have to trust the information which is in the data which is available in the literature or by collaboration partners, and then build your models on that. And also scientists themselves are often aware of this problems. So it's if you look at this questionnaire, it's a bit old already, almost 10 years now, but it was basically the question if there is a reproducibility crisis in the biomedical research field, like around 52% said yes, significant crisis, like 38 a slight crisis, and I don't know, 3% think there's not really a problem. So like overall, if you just ask the scientists themselves, they are aware that there's a big problem. And there are many factors which contribute to this this problematic, and it's often things like selective reporting. So people just report what they like and what fits in their own story. Like it's lots of confirmation bias, and you only publish positive results and so on. Yeah, you have pressure to publish, so you have to basically, even if there is nothing or it's not so important, you have to publish it to have success in the scientific field. And then there's lots of problems like just with statistic and unavailability of code, data, and so on. And there's also like fraud, and but often this is okay for most of the things. Like it's more like structural problems in the system than actual like malintention of people. And there are some efforts to quantify how big this problem is. There are some interesting studies from psychology, and the basic idea is, okay, we take like just 100 publications, which are all in the highest psychological studies journals. So that's basically the high impact publications of the field. We select 100 of them, and then we try to basically just do the same experiments, basically the same questionnaires or the same behavioral things, and we see how much of that we can get out of that. And then you see, if you basically do that, like 97% of the original work is statistically significant, and only 37% of the replications show statistically significant results. And you see this, for instance, like in the here in this plot, where you see the replication effect size and the original effect size, and also the p-values, and you see most of the there are much more there are many non-significant results in this reproducibility effect, and especially also the effect size halves. That means basically, okay, often you do like I have group 1 and group 2, and I'm interested in how is the difference between the means and stuff like that. And you get a certain difference. If you would do a replication, you often get only the half of the effect size. So, and this is often because, okay, there's a lot of I don't know, p-hacking and selective reporting and so on and so on. But generally, like the effects are never as big as you as originally reported, and often it's also like very they're often not as significant as originally reported. This is bad, and you see this also like in a for instance, in the plot of the p-values, and if you look at the original studies, it's basically every p-value is below 0.05, and this is basically the p-value distribution if you do the reproduce, and it's you can see that basically, and it's more like guesswork. It's not like if you take this as basically the truth, and then you try to redo it, and then you see, okay, man, of this truth, there's I don't know, 20-30% is still significant, then that's not really good. It's more like, okay, if you have a fact, then the chance is 1 in 5 that it's true, and this is not very good if you want to build a computational model based on that, or do any science based on such information. Okay, sorry. There are also industry efforts to measure this reproducibility. There were some works by Amgen and Bayer, also again some time ago, but it was more like, okay, basically I try like to reproduce clinical findings, and of course the pharma industry is really interested in this preclinical results when you see like, hey, we have a new cancer medication in mice and in cell lines and so on, and but overall it's like very not as good as we expect, and basically 2/3 of this target validation projects are basically halted because they can't have results, and they see basically highly inconsistent results in 2/3 of the papers. And this is probably also like what you see, like around half to 2/3 of the results are basically not reproducible, and this is really a big thing. It's not like, okay, there's like some outliers which are not you can really trust them, but a lot of this preclinical research is like basically not very good. And there are many problems. Often we have just very poor study design, low sample number, very low statistical power of the methods. This analytical flexibility, it's more like you have a data set and you look at the data set until you find something, and you do basically hundreds of tests and so on. And this is just normally you have to account for this testing and do some p-value correction and so on, multiple testing correction and stuff like that, but nobody's doing that. In the end, they just say, hey, I found something and this is the only thing I looked at. And then, but that's of course not the truth, but they went through the data set until they found something, which is a nice story. And so you often have to basically you have to take all these p-values with a grain of salt. There's a lot of publication bias, which is also a problem that there are often many negative and positive results, but only the positive results are published. And you can imagine if you then have 5 papers who have positive results and 5 papers who have negative results, and only the 5 positive are published, then if you go through the literature and do literature research, you basically, it looks like, okay, there is a strong effect. All the publications show it has something, but the 5 who said there is nothing, they were never published because it was negative data. And yeah, lack of data sharing contributes to this whole problem, because basically we can't redo analysis, and we can't put data together to do better analysis. Yeah, there are some studies, and there's obviously a lot of okay, I don't know, 85% of research is waste because often it's like badly designed, not published, poorly reported, and so on and so on. So there's a lot of discussion in the field. It's overall like a lot of the research we're actually doing is probably not very valuable. Okay. One solution to this whole problem is basically the idea of open science, which is the idea to provide free access to the results of your scientific works, and this free access has multiple layers. I would say it's basically the publication should be open access, so that people can actually read it, and the data and content of your work should also be open access, so anybody can reuse it. And this makes somehow sense because most of the research is really paid by taxpayer money, and so the results of that and the data generated by that should somehow be public good, or at least be reusable. And so it overall has a lot of advantages. Okay, basically other people can reuse your findings, build on that, and also people who normally don't have much money and are in part of developing countries, and small universities can have access to this information. Overall, open science has multiple parts. I would say one part is really the open access, like providing all the publications freely and open. The other part is open data. This is especially for computational modeling interesting, because we often use this data, and for us, of course, it's much easier to access data sets in some standardized formats than to go back to publications and try to retrieve information from the publications. And also important for the computational part, that you want to provide your model code and your model descriptions and all the source code as open source and accessible. Because in the end, you want to reproduce not only the data, but you also the whole analysis, which created most of your results. And so this different building blocks come all together to open science, and at least it's not solving the reproducibility crisis, but it provides at least some means to better evaluate it and to tackle certain problems within that. Okay, and so what is reproducible research? One definition I really like is this here by David Donohoe from 1998. And it's basically reproducible research is an article, or what he said is an article about computational science in a scientific publication is not the scholarship itself, it is merely advertising of the scholarship. And the actual scholarship is a complete set of instructions and data which generated the figures. And I think this is a really nice concept, and it means basically that all these publications you read or publications and text you see online, it's not actually what the scholarship was and what is the research outcome of things. It's just advertisement. And then this is also how it is often, because people are just writing a nice story and writing things together, and they want to tell a certain story part and want to sell their point of view and basically make advertisement for their research and for their results and so on. And what you actually have as results is often like the data sets and the experiments that were done and the protocols or the computational models or the analysis code and so on. This is actually the things which are the outcome. But this is often strangely, you often don't find this in publications or associated with publications. So and this is like somehow really strange in scientific research, that the actual outcome of all of science is basically never made available. It's more like, okay, everybody's writing advertisement about what they did, but they never, well, not many people show what they actually did. And this makes it, of course, this makes it complicated if you want to build on things and build computational models, because it's like you read through a lot of stories and a lot of text and so on, but you have to really get to the actual data and to the actual figures and to the actual information to extract that. And so what means this for computational model and computational science? It means basically the practice of distributing all of the data, software, source codes, and tools required to reproduce the results discussed in a publication. And this means like often where we're coming in here is like at this part. So there's often measured data or analytic data, which is, for instance, pharmacokinetic data and pharmacokinetic parameter, which are calculated from the pharmacokinetic time courses. And then we have computational models, which use this data and analyzes code, which brings together data with the computational models. And from that, we generate with some often called presentation codes, actual figures, tables, numerical results, and then we put this into an article. And so the actual results is basically this part here on the left, and the article and the text is more on the other side. And there, of course, there's not only a yes or no, but there's some spectrum. There's basically a publication on the one hand side, and then you can go towards more reproducibility by providing basically more information and more data, and optimally in a structured and easy accessible way. And then there's another step, which is replication, which is actually somebody really doing, trying to redo your work. So in a certain way, reproducibility is more like of a minimal standard, and it shows more like that that what you did is giving the same results, and somebody else could, with all your information, get the same results. Yeah, importantly, that doesn't mean it's correct what you did. Still, your code may have bugs, your methods are poorly behaved, your experimental artifacts, you have problems in your data, and so on. So it doesn't mean that everything is correct, but it means more like if you provide all the means and all the methods and so on, people would get to the same conclusions via the same methods. Replication is much more stringent, and it means more like somebody else is really sitting down and trying from scratch to do what you did. And this often means like for computational models, like re-implementing the model, redoing the analysis and so on, or using different software and so on. For an experiment, replication means basically to sit down and go into another lab and redo the experiments. Yeah. And of course, this is much more complicated. Yeah. But often the problem that arises already in the reproducibility, because it's really difficult to try to do a replication of existing work if all the information is missing to do that. Okay. And one question is how to manage this in a typical research or in a computational workflow. I really like this citation. It's more like, okay, or like, and this applies not only for like building a computational model, but I would say like for doing research in general, or doing something more structured in general. And it's more like you have to think about yourself as your closest collaborator, or your closest collaborator as you 6 months ago, but you don't reply to emails. And it's more like, and it's often very true, even for, especially for longer projects. If you do like a master project or even bachelor, it's often you have after 2 months you have no clue what you did at the beginning. It's often because you didn't document your things well, and it's unclear what you did exactly, because you just forget what you did, and it's really crazy, because and then you try to figure out what happened there, and how did I get this table, and how did I make this figure, and oh my code is not working anymore, and so often it's you have to think first about yourself, like in the future, if you want to go back to this project or continue working on that, okay, could you do that and make it for yourself as easy as possible? And if you did that, then basically you already did all the steps that somebody else could also like much easier understand your things. And part of this is to do scientific research as I would say, like organization of data and code. And what makes a lot of sense is that you have all your material for one project in one folder, and that you do like some logical separation, like you separate data and raw data from process data and code, and you write like some actual documentation and of what you did in the file in the way of readmes, and you use something like version control to version your files. And this allows you in the end to have basically one project state and some documentation about the changes, and some clear separation of the logic. And this already helps a lot, like in later for somebody else to figure out, okay, what is going on, and what part of the project do I have to change something, and where is something happening. And otherwise you're ending in this okay, you have 50 revisions and nobody has any clue anymore what happened, and you basically you're just lost. And often you just say, okay, oh man, I have to start from there. I'm just redoing it. It's less work than to understand what I actually did before. And so what we're using mainly for that is version control via Git and GitHub. And you should all have like a basic understanding of what this is until now. That's really a code hosting platform for version control and collaboration. And for us, I would say the most important features are that we have really version control of our files, and then we can, in a collaborative way, work on this project. There are also other nice features, like I would say the issue trackers to manage certain issues. And for me personally, like is it like this work anywhere, and especially offline and so on. And this is like a very nice thing, especially if you have more computers and have a laptop, and I don't know, and often work at different workstations and stuff like that. That makes it very useful. And this automatically, by you being able to immediately work somewhere else on a computer, this provides you like redundancy. And it is not like what often happens, people are doing projects and then their laptop is stolen, or I don't know, their laptop breaks and then the project is gone, or the last 2 months of their work is gone. And by having this in a distributed version control system, you're basically sure that you, even if your house burns down, you have all the information here. So I definitely can highly recommend to use it whenever and whenever possible. A lot of things is also like doing automatization. This is more for I would say it depends on how good your computational skills are. But it often makes sense if you have to redo many things, that you try to find some way to automatize tasks. And this can just be even that you just have Excel templates and perhaps have a few macros or things like that. But basically, if you see you're doing things a lot of times, try to make this somehow easier. And this allows you to often save a lot of time, and often like make the work much more the quality, improves the quality of the work, because you avoid repetitive errors and so on. In the long run, you often will save a lot of time, but of course, this is you have to choose wisely what things to automatize and what not. So reality is often like, okay, man, certain things, okay, man, you just spend more time in automatizing things, but you still learn a lot for different projects. Okay, so how should this data and resources look in the end? One thing which came really up in the last years is like this so-called FAIR principles for data sharing and data access, and this FAIR stands basically for findable, accessible, interoperable, and reusable. And this is the key idea. Okay, if you have like some assets you want to share or like distribute, that you make them findable and accessible and that interoperable, that they can work together and that other people can reuse it. And there are certain steps which can help in doing that. And one way is like basically that you have to share your data, that you provide licensing and attribution information. You use formats others can use and tool can easily read. Where possible, you use existing databases and yeah, you share your code, provide the data, share the data with the code and archive your things and provide methods and so on. So it's more like this, let's say this general way of, okay, providing information and yeah, of course you don't have to do everything here, but it's more like a granular approach. So it always makes sense if you have like some more open and easier computer readable formats. You should always try to lean towards these. And if you have like databases for like, for instance, if you're doing experimental work and so on, you should use like or try to access this databases, because often they already figured out what is a good format to store that. And of course you can easily also get like some data integration aspect, because the databases have a lot of data, and then if you have your data in this format, then suddenly you have a much larger database for a project. I don't know. Can you still hear me, or now I think my computer is gone. Oh, sorry.
We can hear you, yes.
Oh, okay, but now my computer is not working anymore. Okay, so let's see if I can. Probably I have to leave for 2 minutes. I have to restart my computer. Sorry for that. Please stay tuned. I'm back in a second. Okay, sorry for that. Just have to bring the slides up again. Okay. Also, if you have questions and so on or comments, please interrupt. For me, it's always interesting to talk. Like, to say a bit about this stuff. Oh man. I don't know. I think this Zoom thing is really unstable today, but okay. Not good. Okay. You still hear me? Okay, top, perfect. Let's hope this works for the rest of the thing. Okay. Okay. So some lessons, basically like I learned from my own thing, and I think that it's also good. Like the idea of such an XResearch group is basically that you learn a bit about how research actually works. It's okay. You're doing actual research in the project, but I think it's also like good to get insights in how research works and in what is good and what's not working. I have to say the thing is like often things like are really hard at the beginning, and then you don't know even where to start because there's just too much going on. But then it will always become easier, and I think this is like with a lot of things, not only in research but in life. You just have to stick with it, and then it's basically it gets easier, and then often things start even making fun at some point. And most of the time, a lot of these things I represent as things you should probably do anyway. Like you should have like some version control and some backups and some redundancies and try to work in a good and open way, and so that others can build on your work. And it's like I said, even if it's just yourself in a few years continuing with things. It's often like surprising that you often do full circles and you don't realize it like when you're young, but like I don't know, like years later you're like, oh man, I did this this small project, and man, this is really basically I have to do something very similar now, and it would be really good if I could just build on that. And if you lost all documentation and if you lost all files and information, then it's really hard, and you basically have to start from new. And I would say things don't have to be perfect, and often like if you can do in 20% of the time like 80% of the thing. And this is also like I think really important in research, that you have to say, okay, man, I don't spend too much time on small things and so on, but you have to say, okay, I have to get to certain levels and pick your the most important fights or the most important things, and then you will really do a lot of progress. Often there are alternative ways. Basically, I'm presenting here what I think works for me and for many of my workflows, but of course there are alternatives, and there are a ton of tools like to support you. And but you will have to be more aware of, okay, what is available and what you can use, and then you look for the right things. And yeah, and often you should basically start this pretty early in the project, because often you realize in the end, oh man, I should have done like that. So you should try it from the beginning on, like to basically do this if you do a project. Yeah, and so a lot of these things are going directly in this XResearch repository. So lots of this. Okay, sorry, my computer today really doesn't do what it should do. So a lot of these things are directly in the in this repository already. Yeah, I'm really sorry. It seems like this Zoom is sort of really destroying my computer. Let's see what's going on. See, I should see this here. Okay. So a lot of these things are directly implemented in this repository, where we work on the data. Like for instance, things are like, okay, the separation in different sub-information. Like we have, for instance, a data folder, documentation folders, the models are somewhere, and our source code for running simulations and executing things is in the source folder. And there are some readmes for documentation. So there's more like okay, the things are the folders are named nicely, and it's pretty clear where you should look if you look for certain things. And we have this version control. It's pretty clear like what is happening and who is like contributing what, and this makes it also much easier with a project. And then this is not much work. It's more like often like if you if you spend like 5 seconds to think, okay, what would be good file names and folder names, then you saved already the 10 seconds you need to find the stuff next time you look for it. So this is often okay. It's this decision. Okay, like just investing 5 seconds to save some time later on, and then you have often like this makes it very, very nice. And this is especially important for computational models, because you use, of course, computer code to access data and access your files and so on. And the more consistent are you with your file naming and your structures, the easier it is in the end to work with them. Seriously, I don't. Okay, sorry. Okay. So good. This was basically the first part about the reproducibility, and now I will show like a bit, okay, how we can make the models more reproducible, and how can we make this exchangeable and understandable. And I would say for the computational models, the one way to exchange it in one standard format is the so-called SBML, which is the systems biology markup language. And this is a software data format for describing computational models in biology and, say, biomedicine. So broken down, it's a little bit like HTML. It's like basically an XML format, but it describes models instead of like actual web pages. Importantly, this is independent of an actual particular tool, but it's more like general language description of the model, and then different tools and different software and different workflows can be based on this model description language. So it's more like a language to describe what a model is and how it looks like and how the equations are, and then people can use that in different tools and exchange it easily between different tools. This is free and open, and I would say like in a large field of the biological model community, this is like a de facto standard. It depends on what kind of computational models you have. It depends on what kind of computational models you have. But if you have like ordinary differential equation models and process-based models, I would say this is the de facto standard for the field. And this is basically what we're having here. We're having process-based models, and our processes are things like metabolism and transport via blood flow, and we have species and compartments, and this is basically what process-based models are. They put species in compartments, and then you have processes which put them somewhere else, transport them, or modify them via metabolism. So this is like basically the ideal language or description language for such kind of models. There are large collections of models available in SBML, and this allows, of course, the reusability of these models and building on that. One big resource is like the biomodels database, and there are like a lot of curated models and also a ton of uncurated models. And these are most of them are ordinary differential equation models, but there are also logical models and constraint-based models and different formulations like Petri nets and so on. Yeah, like I said, mainly process-based models, and this makes the core of the language. So if you have something which are reactions and processes, you would describe this via this SBML core, but then there are additional packages which allow you to extend the functionality and describe more complex modeling structures, like things are for instance, constraint-based models, which are a certain class of models which are often used to model microorganisms and metabolism of microorganisms. Then you have like support for distributions, which is basically allowing you to define distributions for your parameters of species and so on. You have things like groups, which allows you to group certain components. You have a functionality for layouting things, and there's also some basic functional. Okay. Oh man, no, not sure what's going on. Please just let me know if you don't hear me anymore. The Zoom window disappeared, or I have no clue. Okay. Yeah, exactly. And there's also like a functionality for model composition. So if you want to combine certain models, and this is also for us really important because we basically doing a lot of model composition by combining this whole body models, which describe the distribution via the blood flow of the substances with tissue-based models, like mainly like kidney and liver and so on. So we will have like some tissue submodels for the intestinal absorption of lisinopril, for the renal excretion of lisinopril, and probably some metabolites, and for the hepatic metabolism if this exists. And we want to combine them, and this hierarchical model composition allows us to include the tissue models into the whole body model. There are other important things like unit validation, unit checking, which are really helpful for computational modeling, because you can easily you can have automatic checks if your model structure and your model equations make sense. So it's often like you have a description of okay, a rate of change of a metabolite, and this rate of change should be in the substance units per time. Or you have a maximal velocity, and this should be also of a reaction, and this should be also okay in how many moles can I convert per time unit, and so on. So certain physical quantities have certain kind of units. Or blood flow is like, okay, what kind of volume is transported per time? So this has like actual like unit constraints, and so equations which are describing blood flow should be in volume per time. And I have other things like mass and charge balance, and like I said, you have a wide range of tools from visualization over parameter fitting to simulation. If you want to know more, there's SBML.org. There's a good introduction and good information. So how does this look at the core, and what are these components we have in such computational models? There's a lot of lists, so it's basically like an XML-based format. In XML, you have often like lists, and in the list you put objects of certain types, and this is also how here this whole thing is structured. So you basically have a model object, and on the model object you can give certain name, like basically a name, ID, what is my models, also what are the units of my model, and then you can put certain components into the model. Often these are basically compartments, which is stored on the list of compartments, species, which are molecular entities which you put into compartments and can transport it. And then you have reactions, which are converting species into species. Think about a simple biochemical reaction where A is converted to B and C. This would be, for instance, a reaction which is working on three species, on A, B, and C. And then you can have additional mathematical descriptions, like there's this where is it? List of rules, and these are basically mathematical equations which you put into it. It's basically a rule like, okay, if I define my body mass index is my height divided via the body weight square. This would be basically assignment rule, and then you have like if you have a body weight and a height parameter or variable in your model, you can via this rule calculate the body mass index. So this is more like actual equations which you put in the form of rules. You can calculate one thing from another thing. They have the parameters. Example for that is like, for instance, cardiac output. Species is glucose in plasma. It's molecules and compartments. These compartments are the containers. I'm like, okay, here the assignment rules, and then you have the reactions. There are also additional things which allow which are more complicated, but okay. At the beginning, I think the important thing is here that you get this concept of processes. So the things are transported or converted from A to B, and basically everything which happens in the body you can somehow put into this abstraction layer. And this physiological-based pharmacokinetic model, like of drugs, is often like it's very close to that. So it's most of the time like species or molecules are transported or they are converted. Yeah. Okay. So this is basically how this looks in a way. There are many tools to work with models. Basically, what I'm mainly using is a Python package I wrote, and it provides some abstraction layer around libSBML, which is a library to work with SBML models. it has language bindings in many different languages, among them Python, I don't know, Julia, C++. So basically, you can use these models like in very different programming language. There's also an advantage, like if you're not writing your equations and so on in actual code, but it's basically you can reuse it in different programming languages or in different tools and so on. And it has support for the packages I mentioned before, and a lot of things is like I would say the main focus is really on model creation, model manipulation, and on building models. And this is what we will use this tool also for, like basically programmatically, like creating models. And so here's, for instance, an example. You would just create a parameter, you give an ID, and then you can give certain values, and also describe, okay, what is this parameter? In this case, it's okay. It's inhibition constant of bilirubin for a certain importer. It has a unit, and here are like some provided additional nodes for experimental ranges of bilirubin, which is a molecule which appears in the blood. And this can then be rendered by other tools and nicely be formatted and so on. A second tool is basically this SBML for humans. It's an online report, an interactive report, which allows to upload the SBML model and then provides you like some interaction layer to better understand the model. It has some search functionality. It renders like the equations, and you can get a better feeling, okay, what this model is and what it can do. It has support for many of these things, and especially also like for these annotations. So annotations are important. It's basically a description of what your model objects are and what your model variables and parameters mean. And this also goes in this direction of, okay, we want to do some documentation also for other people and collaborators and ourselves in the future. And there we can basically do this description of what it means. So in the end, like normally you would just say, I need a parameter which has an ID and a value. I don't need all this information here, but of course, providing this information, okay, this is a certain class of parameter. There's additional information, why I put this parameter in the model and why it has this range, and what it means, and what are the units. This allows you much easier to understand and grab the model content. And annotations are basically this mapping of these parameters to actual ontologies. Ontologies are very, it's basically a definition of terms which are ordered in a hierarchy. And it's really, I would say, it's a knowledge graph where you have certain terms, and every term is clearly defined what it means. And then you don't have to write the full meaning of everything, but you say just, okay, my thing I have here means the same like this ID here. And like, for instance, here, like I can say, okay, my ID AACT, it means the same like this definition in this NKit ontology under the name C64796, and then means it's a hematocrit measurement. And so I get basically like a lot of this additional information by just saying, okay, my object here is basically the same like this object, or is, and so on. Another tool I use a lot is like this SBML, and this is basically a graph-based visualization of the networks and the computational models. And this allows like to get a better understanding. Okay, what is in the model, and this is especially helpful for this process-based models because you can just visualize the processes, what is going from where to where, and has also this additional information about annotations and so on. Yeah, perhaps I want to show like one example. I think in the introduction I showed already this Pravastatin model, but it just gives an example of how these models could be distributed, and so what we and could be accessed. So what we're often doing here is providing just Git repositories of the models, and there we have the raw XML files, which is the SBML. Like, for instance, here we have like the different submodels here for the Pravastatin, the liver, kidney, intestine, and the whole body. And we also can put all this hierarchical, this comp, especially at the hierarchical model composition. We can put this all together to create our complete model, which is all the tissues together with the whole body in one file. And then we can either look like at the raw XML. So don't get too scared. Normally you don't look too much at the XML, but it shows you this is okay. Let's see if my sorry, I always wait until this reacts here. So okay, and in the end you see, okay, there's a lot of this list of, like, here you have like compartments, and in this compartment there's a lot of information, and this is in a standardized XML format. Like I said, this is mainly the exchange format. Normally you wouldn't interact too much with this format, but you have the tools which provide you an abstraction layer around this information. But you can see here, like I mentioned before, you have basically this list of compartments and list of species, and then you have the species in there and what they mean. Via this SBML for humans, you could basically download this SBML model and then just upload it here to SBML for humans and get a more nicely rendered view of the same content of the SBML file. In this case, you see easily, okay, this has 6 compartments, 4 species, a handful of parameters, and 3 reactions, and so on. This is basically a small liver submodel, and yeah, and by clicking on the species, you get more information, and here you can already somehow a bit understand what's going on. This is pravastatin in the plasma. You have it in the liver, in the bile, which is the liver can take up things from the plasma, and often it's excreted in the bile afterwards. So the bile is a certain fluid, and the liver is collecting things into the bile. So often, like modified drugs, which are modified in the liver, are like excreted in the bile. And this is also happening in the case for the pravastatin. There's no metabolism, but the pravastatin is just excreting in the bile. And from the bile, it can basically go back to the intestine. And so this is what the submodel is describing. It's basically has this different substances here, like pravastatin in the bile. And then you have certain reactions, and these reactions are mainly transport reactions. You can see here, okay, the pravastatin from extern from the plasma is basically imported into the liver. You have an export. This pravastatin from the liver is exported to the bile, and then it can be further transported via the biliary flux to the intestine. And then you have certain mathematical equations for that here, and you have certain units. So rates or reactions are often mostly in substances per time, and your substances are here all in a concentration in millimole per liter. So it allows you, like I would say, like pretty easy to understand what is in here. A lot of things, like unit definition, ports, you don't need so much. I would say the most important things is often like the compartments, the species, and the reactions, and sometimes assignment rules. But you see here in this simple model, you don't even have assignment rules. Yeah, if you look in the full model, of course, this is much more complex. This now puts everything together. The different tissue models plus, oh, okay, this was the wrong click. This is like the raw SBML file, the definition. And here is like, again, this report, which provides like some abstraction layer. You can see, okay, we have many more compartments here, and so it's basically like a lot of tissue and plasma volumes, which corresponds to the different organs. We saw this kidney, liver, lung, gut, the rest of the compartment, and you have certain say like a bit of dummy compartments, which are membranes, because you want to locate transporters at membranes. So the compartments are not really volumes, but more like areas. So you have sometimes really actual volumes. Other like compartments can also be areas in the case of the membranes, and that's basically what you have in here. And this corresponds to this. Don't know if this works here. No, I have the feeling the browsers, like everything is really super slow. Okay, here. this is basically corresponding to this structure. So this is like I said, like this is advertisement basically for the model. So this looks nice and so on, but it's like for you really difficult to understand what is actually happening here in a mathematical way, and why is it behaving, and how would I simulate that, and how was the import like here modeled, and how does it depend on this and this concentration, how is the blood flow working here. It's more like it gives you a concept of like, okay, what is going on, and it is like more like an advertisement for, okay, what is the actual model. And the real thing is basically giving you this model description language where you have all the equations, and you could just reproduce, okay, the simulations we have, because all the math is in the model and all the descriptions are in the model. Exactly. Okay, so this is basically, you can see this much bigger. You have a lot of species, and now also a lot of assignment rules, like, okay, you calculate certain things like body surface area, cardiac output, so on. Okay, that's not good. That's not documented well here, but okay. But you have a lot of blood flow equations, and you calculate blood flows via cardiac output and fractional tissue volumes and so on. So there's just like a lot of logic in these equations. But if you can look at the equations, you can basically figure out what is going on. Okay. I would just quickly show this other tool. So basically we have this okay, we have this cytoscape here. So if it's a bit small, and we could now just download the model. For instance, here I go to the raw XML file, the SBML file, and then I could just download the file. So this is like basically the file. I can just save it here under, okay, I save it under downloads, save, and then I could open it, for instance, in another tool, and this shows just basically the interoperability of the things. If things would react, yeah. Sorry. I think the recording is really freezing the computer all the time. Okay. Okay. This looks good, but okay. Fine. Okay. Let's say import network from file, and then I would just open this file. I just download it. If I find it. This is from here. Yeah, this Power 13 model. And then I can basically do some network visualization. Let's hope that this works. Did this work? Yeah. Okay. Yeah, and then you can see I can look at the species reaction graph of this same model. So this is the same file. It's just a different tool which loaded the model, and it provides some different abstraction layer. This SBML for humans, it provided more like a report-like interaction with this model, and here you can see more a graph-like action. And this is already pretty close to what we saw in this in the overview figure. You have things like, okay, here the venous plasma volume, you have like intravenous venous plasma volume, you have some intravenous injection of the whole thing. You have like some transport, like for instance here through the kidney. So like here, if you have here the arterial blood is, okay, come on. the arterial plot is okay, the arterial plot is going in here, and then it's going through the kidneys, and then it's going here on the venous side. Sorry for that. Okay, but overall, okay, you could interact with this network and understand, okay, what is happening. And most of the things you see here is like either like a transport, or here, for instance, in the liver, you can see this liver submodel where you have like where you have import in the liver, you have this export to the bile, and then you have this export to the intestine. And this was the intestinal submodel I showed like in the other report. And this is here you see it in a graphical way, and it's often better to understand, okay, what is transported from where to where. We also have some more complex views where you can see how parameters and things are connected, but this is more like, I would say, some advanced thing to interact with it. Okay. Okay. So yeah, definitely some tasks which we should do. Okay, if you haven't, like from some people I already saw that they contributed to the GitHub repository. If not, really join this repository and add your information to the repository. You can basically see here on the authors, you can see basically Ray committed this I don't know, 3 hours ago or so, and we see like already a few people in here, but overall, of course, we need a few more. So looks pretty good, but basically everybody should be able to contribute here, and so that we see all the authors. And if you manage that, then you already, I would say, pretty set up for the course, at least like from this version control part, yeah. Okay. Other tasks, I would definitely say, like, try to set up the Git. You probably did this via the GitHub desktop. You could clone like basically this Pravastatin model, and I would recommend like that you basically interact a bit with the model. What I showed right now, that you try to get the reports, look at the XML files, get a feeling, okay, what is SBML? Perhaps visit the SBML webpage, and I can recommend like reading this 2 publications. These are all like pretty high level, doesn't take too much time, and you don't have to read every word perfectly and so on. So it's more like, okay, I think it provides a good high level overview, and it helps you a lot to understand concepts I will use when building the models. So I'm not every time explaining what is a parameter and what is a reaction and so on, but you should basically get a basic knowledge. Okay, what is SBML? What are the objects in there, and what do they mean, and what can they represent? And then this helps you afterwards to understand, okay, now we have, this helps you in doing this modeling abstraction layer, because you have on the one hand side the biology and the medical aspect, and we have to put it into this we have to put it into this modeling language. We have to do this translation, and this is, I would say, this is the main modeling process, like finding the right abstraction layer and translating from this, okay, data and knowledge level we have about a concept into, okay, now I want to have a mathematical abstraction of that. And for that, you have to basically speak somehow both languages. You have to understand, okay, what can I do in the model? And then on the other hand, okay, what is happening in reality? And so, okay, how would I map this on each other in a way that it still makes sense? Of course, you do like some assumptions and so on, but you have to basically understand a bit both concepts to be able to do this process, or at least to follow the process. I think we have like still a few minutes. So what I would do is basically just showing, okay, how we would build such a simple model and how it will look. So I will just close most of the things here and hope that the computer is a bit faster. Okay. Oh yeah, this. Please close.
Matthew asked a question.
Yeah, please ask.
where can we find the slides? Are they available?
I will send basically after the in the evening I will send an email, and then you have all the slides and also the recording. So it's I can probably
Okay,
I can
sure.
put it now in the chat, but I
no worries. Okay, I can wait for it. Thanks. Just so I know like which are the readings and stuff.
Okay, I have to log in. I will send it exactly, and then you can I send it after the lecture. Okay, basically what we have a few minutes time. What I will show is basically how we would build such a model. Basically a very primitive model. No clue. Perhaps we will reuse this for the course, but basically just to get somehow started. Okay, the part I'm not showing now is that I basically have to install libraries and then basically set up a like some environment to develop. So what you have to assume now, I installed the library, this SBML utils and like some other libraries, and now I can create the models and just can press a button and the models are created if I wrote the code. So this part you definitely have to set up, but there will come instruction for that. And I think parts are already in the README also how this is working. And so okay, what we would do is basically build a very simple model, and for that we would okay. Let's see how things are working. First thing is always like it's good to like basically pull the latest changes before you do anything. That's always okay. That's pretty good. Otherwise, it's always a good practice to try to do like when you start working, do some synchronization with the repository, upload files which are still lying around and synchronize, so that you know at the beginning, okay, I'm on the status to which the repository is, and it's not like they say that the longer you wait, like in merging things and bringing things together, the more likely is that there are merge conflicts and problems with other people. So we already try to separate work, that hopefully most people work on separate files somehow. But of course, just try to do often like the synchronization with the repository, and then we avoid like conflicts between different file versions. Okay. So what I would do is basically I would create a new Python package where we develop the Lisinopril. You see Lisinopril things, and I would add basically some subfolder which I call models, and there we would develop the or develop the models in this part. So new Python package, I just call this models. And then we would do some general settings just in this init files. So it's a Python files where you can, which are loaded if you load a certain package. And we just add a few file paths, and it's more that we are defining where our paths should be. And in our case, we want basically know where is all our lisinopril data, and where are our lisinopril models. And okay, sorry, I forgot the. And we want to know how is our final flat model called. In this case, I don't think we need this yet, but this basically we just want to say, okay, our lisinopril path is basically this folder here, and so we can always know where things are defined. Okay, and what we would do now is build a simple kidney model. So here this is already more complex, but we would basically build the simplest possible kidney, and for that we just can generate a file. Okay, let's just generate a file and call this kidney model or model kidney. And here we would define our kidney model for the ACE inhibitor, lisinopril. Okay, The big problem is, okay, we have no clue about lisinopril. We don't even know, okay, man, is this excreted via the kidneys? Is this metabolized? Are only metabolites excreted? But we have no clue. So for now, the main problem is, okay, we know, or I know how to build the model, but I have no clue about the biology and so on, because we basically didn't do the data curation yet, and we didn't look at the data, and we didn't do literature research and so on. So we don't know much about what is happening. But the simplest assumption is, okay, lisinopril is not metabolized. Okay, we take the tablet, it goes through the body, and then it's just excreted unchanged in the kidneys. So this is so basically we have no clue. If we do a first guess, this would be okay, something which could happen. Okay. So the first thing which we have to define is basically that we have to do some imports, and there we import from this SBML utils. We define certain functions. We basically import the factory and the metadata. And then to create a model, we basically we just define a model object and create a model, and then we give this model a certain ID, and this is in our case lisinopril kidney. And that's basically it. So we have a model, and then we just have to call some code to create the model. But this is a really boring model because we sorry, this is always freezing. So for me, it's a bit complicated to type here. We would create the model, and then the model is pretty boring because we don't have anything in the model. It's just an empty container which doesn't have anything in this list of. And to create the model, we would just call the create model function, say which model we want to create. So here, this point, I just call this model kidney. I just give a different name for this variable m. Here, I just want something very short because we have to access this variable a lot, and here I just reassign that. Okay, no worries if you don't understand that in detail. We just want to here say, okay, what do we want to create? We want to create this model kidney, and we have to say, okay, we want to create this somewhere, and we want to create this in a certain folder, and we want to create this in our lisinopril model folder. So this is what we defined before. This in it, we said, okay, in this folder, we want to write all our models which we create. And so we have to give this a name, and we give this a name, the space path, and then we use this ID, which we defined, and give it an XML extension so that we know this is an SBML. And that's basically it. At this point, okay. Oh, come on. We can just execute this this file, and then we will see some output. And here, what we see is basically a create SBML part, and then we see certain things like warnings. Certain information should be set on this here, but overall we see like mostly green, and it says, okay, we created this file here, and it's a valid file, which is pretty good. And then we can of course look at this file. And this is our first SBML model. This is pretty boring. Of course, we just created the container. We didn't put any species or compartments in there. We didn't put any reactions or the processes in there. So this is just, okay, an empty container. And now we have to like add compartments, add species, add reactions, and do some abstraction layer. So this is, I would say, the simplest model. And so what we have to do next is adding some compartments. There's a lot more information which we can add, but I basically start with the minimal information, and then step by step we can add more. Like for instance, here we want to add multiple compartments. So we would add one compartment, which is our we call it v_extern. We give it a certain value. In this, it's just 1.0. And for now, we give it also a name and call it plasma. And this should be pretty good. So we have this here. And so the kidney, what normally is happening, it's taking the blood, which is going through the kidney. It's doing some filtration process, and then part of the substances are filtrated in the urine. And so we don't model this in detail, but we're just saying, okay, our kidney model has somehow access to the plasma compartment, which is associated to the kidney, and it will also have like access to a urine compartment. This we can call urine, and this is our urine compartment. Okay. And then we have two compartments in there. Okay, this is still a bit boring because we just have compartments. And so we also want to have species in there. And then we would add species to our compartments. So we have species, give it again an ID. And in our case, like we would use lisinopril. We need some short name for that. So we would just call it lis. And give a suffix of ext, and this is it's basically located in a compartment. And we have to say, okay, in which compartment should this species located? And in this case, we say, okay, it should be in the external compartment, and so on. And we want to also say, okay, we want a second species which should be located in the urine. So this is our lis in the urine. This is the second species, and this is located in the urine compartment. Okay, so this is two compartments, and we put two things into the compartments. It's still okay. It's more interesting than the first version of the model, but it's still a bit boring because nothing is really happening. So it's nothing is converted and so on. So it's yeah, nothing is really happening in the model from a mathematical point of view. But it's more like if you put like something in the urine in the beginning or in the plasma, it just will stay there forever because we don't have any conversion processes or transport processes in there. And so what I'm do now, I'm basically accessing the results of this model creation, and I call directly a visualization on that, so that we can see what is happening. For that to work, I have to restart this cytoscape thing in the background, so that we can directly do the model visualization while we create it. This is basically this part here. Okay, and then we have this here, and then we can oh, come on. And then we can re-execute here our model building. Sorry, this is really slow today. Normally, so. And then we would rerun it, and then probably would either get like some errors. And you can see, okay, certain things happened. We saw some visualization on this part here. And what you basically can see in the species reaction graph, we just have we created two species, like one in the urine. Should be able to see this here. This is the urine, and the other one is an external compartment, but nothing is happening. They're just located somewhere. You can see this also in this bit more complicated view where you see, okay, these species are associated with these compartments. In this model creation part, you saw like a lot of warnings, and this is more what I meant, like there's a lot of feedback and validation. So we did really a minimal model. This is something which you could simulate, but there's a lot of things which are missing and should be done, and certain things which are probably not very good. So for instance, we get a lot of unit consistency problems, and this is mainly because, okay, the units, we didn't provide any units on anything, so units can't be checked. Overall, these are mainly warnings. You can see this by being the results here in orange. So if it would be red, it's bad, then the model is broken. If it's green, everything is good. If you're orange, you have a lot of warning, and it's basically like not best practice, and units are missing, and so on. And we are somewhere like, okay, we are on this level. Okay, this can be simulated and so on, but it's not a very good model. just to demonstrate the last thing, we would just add a simple reaction. And the reaction which is normally happening is like we have to somehow put this abstraction of, okay, the kidney is filtering substances from the kidney is filtering substances from the what's going on here? Come on. Oh man. It's filtering substances from the plasma into the urine. And so we would put this into a reaction and just add a reaction object. Again, we give just an ID. In this case, we would call this lease excretion. This is basically I just give a name. This is lisinopril renal excretion. This is basically what this process is doing or representing, and it's a strong abstraction. We're just putting this into a simple equation. We have to say, okay, what is this this reaction like actually doing, and what metabolites is it modifying? In our case, it's taking the lisinopril extern and it's converting it into the lyse urine. But converting, transporting. Okay, it's basically taking one molecule of this here of our plasma and putting one molecule into the urine. This is what this reaction is doing, and for that to actually be mathematically simulatable, we need a rate equation and just basically saying, okay, how fast should this be? And how fast this normally is, it depends on some rate constants, and often it's basically something like mass action. So meaning the more we have of a certain substance, the more it can be exchanged. So in the end, there's a simple product. Okay, the higher this rate is for the excretion, the higher it is, and the higher the concentration in the plasma of lisinopril is, the higher the excretion will be. This would be now invalid because we never defined actually this parameter. So we could either define this more globally, or we can define it directly here on the reactions. Okay, this is reacting. So we would put basically the parameter here, a local one. So we have to define an additional parameter, which is our lyse X K rate, and this needs it needs at least some value, 1.0. Okay, things at this point we're getting a bit more mathematical. You can see, okay, we already have concentrations. We have like some weight parameters. So at least we should start and also set initial values for that. So in our case, we would say we have a certain initial concentration of in our case 10. That's basically, I don't know, we don't have units, so it's, I don't know, 10 potatoes or whatever. So yeah, but it makes sense to have units. But for this simple example, it's not so much. And in the urine, we would not have anything. So this would be a simple model where we say, at the beginning, we have 10 of our units in the external in the plasma and nothing in the urine, and then our process should just put this from one compartment into the other compartment, and then we just check if we get at least an orange model. So there's a lot of warnings still, but this is still orange. And if you look at the process-based view, we can already see, okay, now we have actually a connection. And we said like, okay, this lyse extern should be 10 at the beginning, and then there's a certain rate with which this lyse extern is converted into the lyse urine. So if we now start simulating that, in every time unit, like one of this rate unit is like converted from the lyse extern to the lyse urine. And the actual weight is depending on this, our weight constant, and on the concentration of the lyse extern. So the less lyse extern we have, the less this is converted. So this looks pretty good. Basically created a simple model. And now we could, if you're lucky, like even simulate it. I have to see. So if my computer is just dying, I'm sorry for that. In this case, I would use Copasi. This is not something we would use in the doing the course, but it's more like a nice tool if you want to look at a model and do simple simulations of the model. So in our case, we would import the model, which we just generated. So we generated this somewhere in our XResearch repository. And this was written into the source XResearch Lucinopril models. Oh no, not here. Oh, I have to see where this was written. I should have paid attention where the base path is. Yeah, it's probably written here in the models. Yeah, and you can see here this Lucinopril model was generated here, and we can just import this model now in this tool. Okay, there's some warnings, but overall we can have a look at the model structure, and we can see, yeah, we have our two compartments, we have our two species, we have our one reaction. So the things was basically exchanged now, like from using a totally different tool to look at the same model, seeing certain rates and so on. And here we could even do like some simple simulations for that. So here, for instance, we could run a time course and just say, okay, we want to run this now for, I don't know, 100 time units and create a certain amount of points. And then I would just use this output assistance, which says, okay, I want to create certain plots for the interesting things in the model. And then I could say, just run this and run a simulation. Okay. In this case, okay, that's I probably use it a bit slower time. So you can see, like I just put in some initial concentration of 10. And what we can see, what in this model is happening over the time is, okay, the things, basically our Lis extern, which is the red curve, it's just getting less, and it's getting less because this process of renal excretion is putting it into the urine, and at the same time the urine amount is going up, and it's going up to 10. And like with this parameters and so on, it takes around 10 of this time units. We didn't define any time units, and we didn't define any units for this here, but overall it shows like 10 of our things are converted per time unit to this and that. And then that's basically that's basically it. So we put like some general model definitions and put some equations on that, some initial conditions, and then we can run some simulations. And of course now you put more things into the model and try to find the right abstraction layers, and then you can start to simulate more complex things. And then combine that, of course, if you now would have a whole body model for the circulation of lisinopril, and would have a submodel for the absorption of the lisinopril tablet, then we could already couple it to this urinary excretion of lisinopril and just see, okay, does it make sense? Can it simulate and so on? Okay, I would say lots of details. Like, I think the important thing is like just have a look at them. Basically now I will just commit that, so you can already have a look at the code here. This is all in here. You could try to get this running, rerun the model. You can do the visualization with the cytoscape I showed. You can do the create this report I showed before. So there's a lot of things you can already do. You can also, of course, yeah, fork the things or manipulate it. It depends, okay, on your level of like, if I say words you don't understand, then probably don't try it. If it makes sense, you can try it. It's like I have to take you somewhere from the people who want to do more, like who already know the programming part, like the basic could already start playing a bit around and trying to understand that. They could also use look at the SBML utils and at the examples to better understand what these models are. In general, I recommend like reading the SBML papers and looking at the SBML models I showed today. Okay, and then now important thing, if you did some work in the end, you should always like commit the things again. Okay, sorry. I don't know. I probably have to. Oh, man. Sorry, my computer is freezing all the time. I would say, okay, I will just commit the changes. From my point, this was the main things I wanted to show today. Yeah, if you have questions, please let me know. And also, if you have feedback, just let me know. This is a lot of things I'm not expecting you to learn the things and so on. It's more like, okay, I want to provide you the information, and you should more actively just try to interact with that and try basically you understand where your knowledge gaps are, and you should try to fill them and basically read up on the parts which you didn't understand so much. So it's not like, okay, you have to do this task and so on. So it's more like, okay, you should try to get up to speed, and the things you didn't understand, okay, just ask or try to interact with that. Okay. So can you still hear me, or am I gone now?
You're still here.
Ah, okay, yes, the top. I thought I have to throw everything again. Okay, perfect. Yeah, no, my computer froze again. So I will just leave it like that. I will commit the changes. I will provide the slides, and yeah, if you have questions, just ask now, or later via chat and or email. Okay. Otherwise, thanks a lot, and yeah, like I said in the email, next week I have to do it 2 hours later, unfortunately, because I have to teach during the whole day, and I can only make it at 6. Okay, I will send around all the information, and then yeah, hope to see you all next week.
Thank you, Matthias.
Thank you.
Thank you. Bye.
Bye.